Hi everyone, I'm glad to be here talking about my thesis, Effect of Host Heterogeneity on Vector Attraction, Disproportionate blood feeding dominated transmission of Plasmodium and West Nile virus. And my supervisors are Jody, Josue, and Laura. I would like to talk about my thesis from 6 parts. You will get to know the background, scientific question, material method, as well as some results and the upcoming works. Here we go to the topic. Nowadays, the transmission and the emergence of infectious diseases has been accelerated. According to the report, the number of range-expanding infectious disease has increased. Human activities such as land use change are the main drivers. In addition, about 75% of the infectious disease is caused by zoonotic pathogens such as Plasmodium and West Nile virus. Avian Plasmodium and West Nile virus are two important study models. In nature, avian Plasmodium circulate between birds and mosquitoes through mosquito bites. They accomplish the asexual stage in birds and the sexual stage in mosquitoes. For West Nile virus, it can be transmitted to bird as well as human and horse. However, human and horse are dead-end hosts. This means they cannot pass the virus on to other biting mosquitoes. So birds are the major reservoirs of these two pathogens. Basic reproductive number, as known as R0, is the expected number of secondary cases caused by a single infection. And here's the value of some well-known infectious diseases. The higher the number goes, the faster it will spread. When R0 is smaller than 1, the infection will die out spontaneously. But if R0 greater than 1, the infection will be able to spread. And here's the scenario when R0 equals to 2. It will be transmitted rapidly if every individual is equally susceptible. In nature, however, different hosts are not equally susceptible to mosquitoes. In other words, hosts and vector are not randomly interacting, so R0 may be altered by the varying host-vector counter rate. However, previous models based on R0 tend to ignore the impact of inter or interspecific difference in hosts. We may ask, is it wise to ignore the impact? In extreme cases, 20% of the host populations contribute to at least 80% of the transmission potential, which may cause super spreading events. Obviously, these 20% will overweigh those 80% in epidemiological studies. In our case, if mosquitoes feed disproportionately on infected birds, the transmission of infectious disease will be driven due to the increasing host-vector counter rate. And here's an example. A study on the blood meal composition showed that some mosquito species feed predominantly on birds. And coincidentally, these bird biters were at a higher risk of transmitting West Nile virus. Among others, heterogeneity in host traits may largely determine the disproportionate feeding by mosquito. And here's another study. The left column is the relative abundance of host species, and the right column is the feeding proportion of blood meals. It's clear that some species, such as common blackbirds, were bitten more often than expected from their lower abundance, while other species, such as common starling, were bitten less in spite of their higher abundance. However, previous studies used to assume that host traits did not affect mosquito attraction. As a result, the impact of host trait heterogeneity on the disease transmission is poorly understood. Thus, incorporating host trait in our study and identifying what traits are the causes for the disproportionate feeding will be of great importance. So our scientific question is, How does heterogeneity and host trait affect the feeding patterns of disease-transmitting mosquitoes? To answer this question, we developed 5 goals to achieve. The first, understand what cues mosquito may use for blood seeking. And second, figure out the role of host metabolism in mosquito blood seeking. Third, test the effect of malaria and infection status on mosquito blood feeding. Fourth, test the attractiveness of host morphology to mosquitoes. The last, figure out the factors that determine the outcomes of host competence. To fulfill our goals, a variety of materials are needed. We will use house sparrow as our study models, not just because it's a ubiquitous species, but also because it usually has a high prevalence of infection. And common house mosquito will be the primary vector as it is competent for transmitting multiple diseases. And we'll use Plasmodium as the main pathogens. Now we are approaching our goals from 3 paths. From the general path, we'll review all the host traits that may be used by mosquito. From the interspecific path, we will experimentally test the roles of host metabolism and infection status in vector attraction. From the interspecific path, we'll use comparative method to test the effects of host morphology and immunology on host vector attraction and host competence, respectively. So there will be 5 chapters in my thesis, and here are the titles. Here are the titles for the chapters. Chapter 1 for the review. Chapter 2 for metabolism. Chapter 3 for host infection status by Avian malaria. Chapter 4 for the morphological traits, and the last one for the host competence. And here we go to the chapter 1. First, we'll try to answer the question, what kind of host trait may serve as cue for host-seeking mosquitoes? And we'll examine the roles of morphological traits, and behavioral traits, and physiological traits in the process of host seeking. Second, how this cue works in different distance, time period, and other conditions. You know, the relative importance of this cue may be affected by the distance between host and mosquito, by day or night period, and by host color contrast against the background. In chapter 2, we'll test the effects of host metabolism in mosquito blood feeding. We first prepare unfair female mosquito by field sampling of larvae and the artificial rearing of mosquitoes. And every mosquito we use in the experiment has been identified to species level. We also collect the house sparrow in the field. Their individual profiles, such as morphological traits and blood status, were established. And then a pair of birds containing a male and female with different metabolism was exposed to mosquitoes during the night period. And 15 trials with the same setting has been conducted. We measured the resting metabolism as oxygen consumption rate before and after the experiment. And each pair of birds was exposed to average 190 mosquitoes. In total, over 2,800 mosquitoes were introduced in the cage, and nearly 800 of them were fed. Then we processed these fed mosquitoes to molecular analysis. And basically, 30 engorged mosquitoes were randomly picked out from each trial for the analysis, as we cannot afford to test all the samples. And PCR sexing were first conducted to partially identify the blood meal origin. As you can see here, those 1 banded sample were blood meal taken from male bird only, while those 2 banded samples may either from female or from both birds. So we'll process these 2 banded samples to microsatellite genotyping. And we managed to separate this sample by using 8 different primer pairs. And as you can see from this figure, and this one banded sample was taken from female only. And those 2 banded samples were brand new taken from both birds. And for the remaining sample that without successful amplification, we will perform DNA sequencing by comparing the size of 4 different fragments. And finally, we managed to figure out the origin of all selected samples. Then we calculate the biting rate as a response variable. Here, biting rate for a bird was calculated as the sum of the number of female mosquitoes from a bird and from both birds, divided by the number of total female mosquitoes. By using resting metabolic rate and physical parameter as predictor and bird pair as a random effect, we conducted modeling on the variation of biting rates. And we found that biting rate was positively correlated to the body mass of birds, but negatively to the resting metabolic rate. This means mosquitoes may prefer to bite those birds with bigger body size but lower and lower metabolic rate. Here we go to chapter 3 to test the effect of host infection status by avian malaria. And basically, we were using the same method as we did in previous chapter. The difference is that we performed 2 experiments here. One is the comparison between the healthy and infected bird, and with 20 trials. And the infection status was identified by PCR using primer that targets on cytochrome B gene. And the second one is the comparison between medicated and controlled birds. We medicated half of the infected bird by injecting primaquine, a well-known anti-malaria drug, which can lower the parasite loads after the injection. And the second half of the infected birds was injected by PBS as control setting. And we also checked the parasitemia on the blood smear. Here, parasitemia is the number of infected cells among 10,000 red blood cells, which can be counted on the 10,000 magnification of microscope. In total, we've got a huge number of fair mosquitoes. And then, by using the same method as we did before, and we got some preliminary results here. For the first experiment, we didn't detect any significant relationship between bird trait and biting rate. In addition, there was no significant difference of biting rate between infected and healthy bird. This means mosquito might have bitten healthy and infected bird by chance. For the second experiment, we found that the biting rate was negatively correlated to the medicated treatment. And this means mosquitoes prefer to bite those controlled birds over medicated birds. We may ask, what makes the difference between controlled and medicated birds? So we compare the parasitemia between these 2 groups. And results of PRT tests show that those controlled birds possess higher parasite loads than those medicated birds. Thus, parasite load may be an important factor driving the feeding patterns of mosquitoes. And here we go to chapter 4, dealing with the morphological trait and forage ratio. We got the data of forage ratio for 49 bird species from a published paper. Here, forage ratio was calculated as the fraction of total whole blood meal from the whole divided by the relative density of the host. When the forage ratio is greater than 1, means a whole is overused, but if forage ratio is smaller than 1, a whole is underused. As you can see from this figure, and the forage ratios varies a lot across different species. Some species, such as American kestrel, were bitten more often than expected, while other species like common gecko were bitten much less. Then we collect data for host traits. For plumage coloration, we first divided all plumage colors into 3 categories, and that is highly, moderately, and slightly attractive colors. And then we divided the entire body into 7 parts. And the relative size for color category in each part was estimated by percentage. So the final score for color category was the cumulative sum from each part. And we also collect data for body size from a number of literature. We then built a consensus tree based on 1,000 trees by adopting a 50% majority rule. And here is our tree. And to control the potential autocorrelations between forage ratio and bird phylogenies, consensus tree was incorporated into the PGLS models. However, there was no phylogenetic signal accounting for the variations of forage ratio. And in the batch model for Curious Pigeon, we found that forage ratio was positively correlated to tassel length and slightly attractive colors, but negatively to the bill length. In the batch model for Curious Restaurant, we found that forage ratio was positively correlated to slightly and highly attractive color. Thus, morphological traits can really affect the feeding patterns of mosquitoes. And here we go to the upcoming works. In the last chapter, we will do a comparative study on host competence on West Nile virus. Host competence is the ability of an infected host developing sufficiently high levels of virus to infect mosquitoes. In nature, not all the infected hosts can transmit virus because some species may not tolerate such a high levels of virus. So we are trying to figure out what traits of hosts may determine the outcome of host competence. And we use viremium level as the response variables. Now we've got some data from 77 bird species that have been experimentally infected by West Nile virus. And based on these threshold values that has been established as necessary for infecting mosquito, avian hosts will be classified as incompetent, moderately competent, and highly competent for the transmission of West Nile virus. And some immunological traits such as antibody and white blood cell level, as well as virus lineage, will be the predictors. And we'll perform PGLS modeling like we did before with the control by bird phylogeny. And obviously, this work cannot be done all by myself. I really appreciate the help and support from my group and from the EBD, as well as from my funder. And that's all. Thank you.